Hello, everyone. Happy today. In this chapter, I believe 45, we get into the endocrine system and look at how chemical signaling occurs within organisms. Now, this lecture will be covered live in class, so that way we can have a little bit more interaction with students. But if you happen to miss lecture on the Zoom call, here you go with the information. So, the endocrine system, again, you have glands throughout the body that release chemicals that are detected by particular tissues. So, the thrust of the idea is cells need to coordinate their activities. You're made of trillions of different cells. You have 200 to 300 different types of specialized cells, but they need to work in concert with one another in order to keep the organism functioning. So you have all these different cells doing all these different jobs, but they have to be able to communicate with one another in order to produce the right chemicals or the right reactions needed for the organism. So you need communication systems between cells. So there are two primary means of this, and there'll be different chapters that we cover. The first is the endocrine system, and the other is the nervous system. Now, with the endocrine system, again, you have a secreting cell, typically glandular tissue, produce and then release a hormone that travels through the bloodstream. So one of the hallmarks of the endocrine system is that the chemicals travel through the bloodstream. So you've got 60,000 miles of blood vessels in the body delivering materials very close to cells. So those hormones travel through the bloodstream, then they go through the interstitial fluid, and then they'll dock on receptors either on the inside or the outside of cells, and then that allows the cells to produce some sort of response. Now, this process tends to be slower. It can take minutes to hours to months for processes to occur, but the duration of those responses tend to be a little longer lasting. Well, let's contrast this with the nervous system. The nervous system communication happens in milliseconds, right? Incredibly fast, but tends to be shorter in duration. So think of fingers playing an instrument. So fast response, shorter duration. And rather than having the sort of circuitous route for chemical signals, there are specific pathways or routes dictated by the travel of axons, the extension of the nerves. So the nervous system uses electrical signaling combined with some chemical signaling in order to elicit specific responses from specific tissues. So they're both sort of specific in terms of their targets. Either you have a neural connection or you have chemical receptors, but they sort of travel through different routes. They operate at different speeds and they last for different lengths of time. So in this chapter, we focus on the endocrine system. The nervous system will get its own chapter. All right. So looking at chemical signaling, the general rule of thumb follows what we learned in chapter 11. You have a ligand, a signal that's detected by receptors, either in the cell membrane or in the cytoplasm. You get this transduction that occurs. Usually the first transduction is a change in shape of some domain or region on the receptor, and then that relays a signal through shape change of molecules within the cell, and it elicits some sort of response. Either you switch on an enzyme or switch off an enzyme, you switch on transcription or you switch off transcription, so you're making proteins or not making proteins, but the cell's going to do something. All right, when we look at hormones, there are two categories of hormones, that I would like us to consider. There's going to be water-soluble hormones or lipid-soluble hormones. Remember, like dissolves like, right? So with water-soluble hormones, again, they're hydrophilic. If we look at the structure of these, you'll notice that they are polar. So water-soluble hormones or hydrophilic hormones are polar. And as such, they're not going to be able to pass through the plasma membrane of the cell. So they're going to be detected by receptors on the surface of the cell. So here's your glandular tissue. The hormone travels through the bloodstream, and then it docks on a receptor on the inside of the cell. So it can either switch genes on or off, or again, it can switch enzymes on or off. Lipid-soluble hormones, they are nonpolar, and those can actually pass, through the bloodstream, through the cell membrane, and even into the nuclear membrane. So lipid-soluble hormones tend to switch genes on or off. Let's see. So let's look specifically at the water-soluble hormones. Again, if they're water soluble, they are polar with like dissolving like. They tend to be protein based. They're detected by receptors on the surface of the cell. And a lot of times, again, through signal transduction pathways, they switch enzymes on or off. Now, to stop the signal, what do you have to do? Well, you just have to remove the ligand from the receptor, right? So their changes tend to be shorter lasting. So to contrast endocrine system and nervous system, think of like seeing a bear. You see the bear, you get startled, and again, don't run, never run. Let's say your body gets all pumped up and filled with adrenaline, right? So, okay, let's say you run, and you take off. And so for a short amount of time, you're going to have your arms and legs pumping. You get out of there, you're safe, you're in the car, or whatever the case may be. Your nervous system stops responding to the side of the bear, and your muscles stop contracting rapidly. But think about how you're going to feel. Your endocrine system is still coursing those hormones through your body, and you're going to stay pumped up for a period of time. Okay. Now that may dissipate after 15 minutes, half an hour, and then you sort of go back to baseline in terms of blood pressure and heart rate and that kind of stuff. So your nervous system is really fast, but the reaction stops more quickly than does your endocrine system. So looking at this pathway in particular, here you have adrenaline docking on the receptor. That'll cause a shape change in the receptor. Then this looks like a G protein coupled receptor here. You get the G protein go and dock on this enzyme adenylyl cyclase that causes the production of CAMP. And then CAMP causes the activation or phosphorylation of these different molecules. And you end up activating this enzyme. When you activate this enzyme, then it will cut up glycogen. When it cuts up glycogen, then that releases sugar into the bloodstream. And then you can power cellular respiration to make ATP to fight or flee. I'm more of a fleer than a fighter. So let's contrast this with the lipid-soluble hormones, the non-polar hormones. Because they are small and nonpolar, they can squeeze between those hydrophobic tails and the phospholipids of the membrane and dock on receptors inside of cells. When they dock on receptors inside of cells, they tend to activate expression of genes or switch off genes, and that alters protein production. And remember, proteins, like enzymes, they do the work in cells. So if you produce proteins or don't produce proteins, you're going to change what cells do. And because you tend to be switching genes on or off, they tend to have longer lasting effects. Think of like testosterone and estrogen here. This is looking at testosterone. You can produce proteins that are associated with like the secondary sex characteristics of males, and that can cause like long-term or permanent changes in the body. So the AP curriculum looks at all sorts of specific examples of hormones. So what I'd like to do is look specifically at epinephrine or adrenaline. So whatever they give you on the AP exam, you can tell things through the visual clues they provide. If we look at epinephrine, notice where it docks. It docks on a receptor on the surface of the cell, so we know, without even getting any other information, that this is going to be a polar hormone. Okay, so this is one of those protein-based hormones. It docks on receptors on the surface of the cell. It activates this pathway. And again, what does it do in this instance, in this particular cell? It stops the production of glycogen and then activate enzymes that cut up or hydrolyze the glycogen to release glucose. So again, that's the specific reaction in that particular cell. Now, what we can do is think about a specific for instance. Again, always thinking about what they could do on the AP exam. Epinephrine or norepinephrine, they're also called adrenaline or noradrenaline. So if we look at the image here on the left, there's adrenaline or epinephrine or noradrenaline or norepinephrine. It docks on receptors of the cell. That can do things like make the SA node in the upper right chamber of your heart send out signal more quickly. So it basically increases your heart rate. It makes blood vessels in some part of the body dilate and blood vessels in another part of the body constrict. So you send more blood to your extremities and less blood to your guts to digest lunch. And it causes all these reactions to the body to get it, again, pumped up and ready to go. Well, beta blockers, if we look at them, I just covered it up, I'm sorry. if you look in the bottom left, once this clears out, what beta blockers will do is dock on that receptor and block epinephrine or norepinephrine. And as a result of that, you shut down this signaling pathway, and it can do things like lower heart rate and lower blood pressure by making blood vessels dilate. So again, think of what ifs. Here, this is out of your book, looks at some of the effects of stress on the endocrine system. So when the hypothalamus detects a threatening stimulus, you send signals to the adrenal glands. Those are glands that just sit on top of the kidneys, and they'll release stress hormones like adrenaline or epinephrine and cortisol. So on the short term, epinephrine and norepinephrine, or adrenaline and noradrenaline, again, they're going to cause the breakdown of glycogen and release glucose into your blood. It's going to alter your blood pressure, your heart rate, and it's going to change blood flow through the body. So again, more blood goes to the extremities and less to the guts. Now, long-term effects of things like cortisol, that's what you see in the box there in the bottom right. It can keep blood sugar levels elevated, which can be damaging to the nervous system and the cardiovascular system. It can suppress your immune response with long-term effects of stress. Because you have more sugar in your blood, it's going to increase, it's going to draw water into your bloodstream and increase blood pressure, which is damaging to the lining of your blood vessels. So there are a lot of downsides to prolonged stress. So one of the best things you can do for yourself is, any sorts of activities that reduce your stress, because when you are stressed, you're releasing these chemical signals in your body that cause changes that damage it. So I know it's certainly easier said than done, but trying to do things to reduce your stress is always good for you. All right, here we're going to look at another hormone. It's estrogen, or estradiol is a version of estrogen. There are a few different types of estrogen molecules, and this estradiol is a one that's frequently produced in younger females. So again, what can we tell? Here it docks on a receptor inside the cell. So hey, we're dealing with a nonpolar hormone. As a nonpolar hormone, it can get all the way inside the cell and then activate genes to produce proteins that, again, can influence secondary sex characteristic females. Or this vitellogenin protein is really interesting. It is a protein that is ultimately used to make egg yolks in females and can play a role in making eggs for females. And it's seen in all sorts of different animals. One thing that's, I think, sort of cool about the vitellogenin is its effect on bees. So for the worker bees, it can actually influence their behavior and influence their sort of foraging activity, going and looking for food. Also, it can act as like an antioxidant to protect the queen bee. So the queen can both be long-lived and to be fertile and produce lots of offspring. So that's sort of a fascinating protein related to estrogen levels. Here's something else that's sort of interesting related to estrogen. Again, the shape of the molecule matters, right? Because the shape of a molecule can dock on a receptor and elicit responses. So BPA, it's bisphenol A. It is a chemical produced in plastics. I don't know if you've seen stuff that talk about like BPA-free water bottles. But BPA is an endocrine disrupting chemical. That's that EDC. Because what it can do is it can act as an estrogen mimic. So here you see, naturally produced estrogen docks on a receptor inside the cell, and it can activate the production of certain proteins. Well, what BPA can do is also dock on those receptors and then stimulate the production of different types of hormones. So it can actually alter expression of genes and can do all sorts of things to disrupt the endocrine system in individuals that consume that. So there's been a move. Once people have recognized that BPA can sort of mess with people's endocrine system signaling, they've removed it. Now, BPA, it's found in liners and like canned goods. Again, it's in water bottles. And one thing that can happen is if people heat up plastics, it can start to break down the plastics and leach BPA into the food or drink. So, not running plastics through the dishwasher and washing them by hand can be helpful, or storing foods in glass containers rather than plastic containers, or again, making sure you buy BPA free stuff, can do things like prevent disruption of endocrine systems in individuals. So that's sort of fascinating. Let's see. While we're on the topic of estrogens, estrogen exposure is involved in the development of like breast cancer. Lifetime exposure to estrogens plays a role in a woman's likelihood of developing breast cancer. So what can happen in breast tissue is like what we see here. Estrogens can dock on receptors and activate genes, and on certain cells can stimulate cell division. Now, a lot of times that regulation is normal, but sometimes people have mutations that cause an overproliferation of these molecules and can stimulate excessive cell division, so you get breast cancer. So one type of treatment that has been developed is tamoxifen. So I don't know if you guys have heard of tamoxifen before, but what it can do is actually be an inhibitor by blocking this receptor for estrogen, so that cells in breast tissue don't get that chemical message, and it slows down division of the cells and can limit cell division. So a lot of times after cancer treatment, sort of depending on the type of breast cancer the woman has, she can take tamoxifen and significantly reduce the odds of recurrence of breast cancer by taking this molecule that, again, basically blocks the receptor for estrogen. The last type of receptor we'll look at related to breast cancer here is the HER2 receptor. HER2 receptors, again, are found on membrane proteins in breast tissue and can stimulate cell division. Now, people can have mutations that cause an overproduction of HER2 receptors. And if you have an overproduction of HER2 receptors, then that can cause excessive cell division and, again, tumors or cancer. So another breast cancer drug that's been developed is Herceptin. So what we can see is that Herceptin can join with and alter the shape of these HER2 receptors. And if you block those receptors from functioning, that can limit cell growth. So women who have HER2 positive cancers sometimes can do well with Herceptin. So one thing that's been really helpful with cancer treatments over the years is doctors can find out, what type of signaling pathway is being affected to cause the cancer. And if you can identify that, then you can use specific drugs that work to block cell division. So understanding biochemical pathways is really a powerful way of getting things to happen or not happen. Last topic we're going to look at here is human growth hormone and its impact on IGF-1. It's called an insulin-like growth factor. So the pituitary gland hanging off the base of the brain can produce growth hormone. That growth hormone can stimulate the liver to produce this insulin-like growth factor, or IGF-1. And then IGF-1 has all sorts of impacts around the body. A couple of big things as it relates to development is causing long bone lengthening and muscle cell development. Okay. Now, one thing that's sort of interesting about muscle growth is the impact of IGF-1. So if a person is doing, weightlifting, or, pumping iron, what happens is the stress of lifting that weight rips the membranes and the proteins. And when the membrane rips, what that does is it sends out a chemical signal that stimulates the release of different molecules like IGF-1. So when IGF-1 is released, what it will do is cause the proliferation of these cells called satellite cells. Satellite cells are sort of wrapped around the muscle cell or muscle fiber. And satellite cells, they have nuclei. And since they have nuclei, you can use the nuclei to make proteins, right? So if you have more satellite cells, that basically means you have more DNA. And if you have more DNA, you can make more proteins. So then you start making more of these actin and myosin proteins, and then that thickens the muscle cell. So when people are lifting weights, what they're doing is basically causing chemical mechanical damage to their muscle cells. That stimulates the release of IGF-1 and other hormones. And then that causes the division of these satellite cells so you can make more actin and more myosin and thicken the muscle cells. And then people get swole, they get big muscles, right? The funky thing about this stuff, though, is like IGF is also linked to activity of the HER2 receptors. So if people have elevated levels of IGF-1, that could impact development of other tissues. So that's another thing to consider with all this, is there's so much interrelatedness between all these different chemicals that if we start messing with one signaling pathway, it can have a multitude of effects on other signaling pathways. So metabolic pathways are all intertwined. Everything in the body is interconnected. So if we can understand these interactions, then we can sort of better understand how to tune things or, upregulate some processes or downregulate others. So it's really sort of a fascinating way to go about understanding the body. All right, we'll pause here. If you have any questions, let me know. But otherwise, hope you're doing well. Thank you.